Good afternoon. Okay, let's move to the new session, which I'm going to chair myself again. And because Pepe couldn't come to the meeting. So the first speaker is Pepe López Barneo from the Instituto de Biomedicina, Sevilla-IBIS. And he's going to talk about mitochondrial dysfunction and compartmentalized alterations of nigrostriatal dopaminergic neurons in Parkinson's disease.
Okay, good afternoon. I thank you for the invitation to talk here today. And I also want to say I'm also very glad, Eduardo Tolosa mentioned already that, very glad that we are meeting in person again after these years without being in contact. And I'm also very happy that the CIBERNED is still alive and doing very well and maintaining the high level of scientific standard that we want the center to have, okay? I'm going to talk about mitochondria, dysfunction and Parkinson's disease, and let me remind you that I'm going to deal with the nigrostriatal pathway, which is responsible for the cardinal motor symptoms in Parkinson's disease. And it is known long ago that classically the mitochondria have been thought to play an important role in the pathogenesis of Parkinson's disease for many reasons. The most important ones are that many of the proteins encoded by the genes which are associated to sporadic and genetic Parkinson's disease, they sound like, for example, Parkin, PINK1, LRK2, etc., DJ1, they are somehow, they interact with mitochondria, they modulate mitochondrial function. And also, as you know very well, some of the toxic compounds, like MPP+, rotenone, used to generate animal models of Parkinson's disease, interact with mitochondria too. Okay, so mitochondria is believed to play a role in the pathogenesis of Parkinson's disease, but the real role of mitochondria in PD is not really well understood. And years ago, I started a project in my laboratory mutating one of the subunits of mitochondrial complex 2, or succinate dehydrogenase. We knock out the gene encoding SDHD, which is one of the subunits anchoring the mitochondrial complex 2 to the membrane, to the inner mitochondrial membrane, and participate in the dehydrogenation of succinate and the transfer of electrons to mitochondrial complex 3. The idea of this project, the original idea, was to generate paragangliomas in the peripheral nervous system, but in parallel, we took a look to the central nervous system because we generated an animal model in which the gene SDHD, coding SDHD, was controlled, was deleted, was a conditional knockout, was deleted by the CRE controlled by the TH promoter, the tyrosine hydroxylase promoter. And you can see, and we look at the evolution, the progression of the alteration in the mesencephalic dopaminergic neurons. And as you can see here, in a normal animal, there is a postnatal maturation of the substantia nigra dopaminergic neurons, here neonatal, P30, etc. And as you can see here in the lower part of this image, in our knockout, we saw a progressive decrease in the number of dopaminergic neurons in the VTA and the substantia nigra. And as you can see here, the effect on the substantia nigra compacta were stronger than in the VTA, as it occurs in other models. But a very, I think, an interesting observation that we saw in this model was that, as you can see here, we saw very early a complete disappearance of the dopaminergic phenotype in the striatum. As early as between P30 and P35, the striatum was already completely, the immunoreactivity to TH was practically gone, and when we measured the dopamine levels was almost zero at this stage, and also the metabolites of dopamine also very much disappeared. And this was very interesting from our viewpoint because at this level, at this time point, that, as you can see here, there were a number of dopaminergic neurons left in the substantia nigra pars compacta, but the striatum was completely not denervated, but the phenotype, the dopaminergic phenotype, has practically gone. And in addition, we saw that another interesting observation in this model, I think, was that when we measure the behavior, the ambulation, the gross motor activity, the movements in the open field of the animals, you can see here, this is the normal animal, here the knockout animal. You can see that between pre-30 and pre-75, there's some decrease in the number, in the distance made by the mice, but this decrease in ambulation, this decrease in the gross movement continues, progresses, even after the striatum is totally denervated, indicating that something else is going on that promotes the bradykinesia, akinesia in this animal model. So just to review, what we learned in this animal model was that the substantia nigra compacta neurons are more susceptible than BTA neurons to mitochondrial complex 2 deficiency, that the phenotype in the striatal axon, this was a model of axon first alteration. The loss of phenotype in the striatum precedes the loss of phenotype in the mesencephalic neurons, and the ambulatory deficits, as tested in the open field, develop progressively even after complete loss of striatal dopamine, as I showed you before. Based on this data, we decided to move forward and to study mitochondrial complex 1, because it is mitochondrial complex 1 which have been classically associated, defects in mitochondrial complex 1, which have been classically associated to Parkinson's disease. This is a classical paper, a seminal paper, showing that, published in 1990, where they look at the tissue from patients who die of Parkinson's disease, and they reported deficiency in mitochondrial complex 1. In the same year, this same group published another paper where, in which they show that these deficits were not just generalized in the whole brain, but was very specific of the substantia nigra, also indicating that these patients who die of Parkinson really have a defect in NADH dehydrogenase, mitochondrial complex 1, in their substantia nigra neurons. And then we decided a few years ago to develop in our laboratory a mitochondrial complex 1 knockout model. What we did here, you can see the mitochondrial complex 1. This is a super macromolecular complex of more than around 44, it's a discussion between 44 and 45 subunits. And 14 of these subunits are maintained, between bacteria and higher mammals. They form the catalytic core, this one here in color, okay? There are 14 subunits, and in this mitochondrial complex 1 function is to convert to dehydrogenase NADH and to produce quinol that is transferred, has the electron that will be transferred to mitochondrial complex 3, okay? So what we did was to look at the quinone binding site, where quinone get the electron from NADH here at this point, and this binding site is formed by 3 subunits, which I say again, they are very well preserved all over the phylogenetic scale, and we decided to knock out the gene encoding for the NDFS2 subunit in order to disrupt the catalytic function of mitochondrial complex 1. So we did a conditional knockout, and when you look at the tissues in which we have removed this gene, you can see that this is a blue native. Mitochondrial complex 1 signal is almost gone. Here you can see it very clearly using antibodies against NDFS2, and this is a Western blot. And of course, looking at different tissues, we could see that the mitochondrial complex 1 activity is practically gone in this animal model, indicating that it is an animal model without mitochondrial complex 1 function, without the NADH dehydrogenase activity, okay? So, and based on this animal, we generated an animal model. Here you can see, we call it MCI1 Park mouse model, because as I will show in a moment, it shows a very progressive Parkinsonian phenotype. And we were breeding animals with, instead of using TH-CRE animal, we use D8-CRE, because D8 is the dopamine transporter that is expressed only in dopaminergic neurons in the central nervous system. And therefore, we generated an animal model which the deficiency, the lack of NDFS2 was focused, occurred only in central dopaminergic neurons. So based on this animal model, what we have seen is that this animal generate a progressive Parkinsonian phenotype, which allow us to do the staging of the Parkinsonian phenotype during time, okay? And what we saw that we saw before in the mitochondrial complex 2 model, that the first sign that we observed in this animal model was early axonal dysfunction. We saw as soon as P30, postnatal 30 days after birth, P30, we see already a disappearance of the TH positive or the dopaminergic phenotype in the dorsolateral striatum. Here you can see here the quantification of the TH expression, where this disappears, where the substantia nigra is still very normal. Still, we cannot see any sign of decrease in the expression of dopaminergic markers in the substantia nigra. So we ask what is happening with these neurons, with these nigrostriatal neurons in which the axons are being already affected. We did a transcriptomic analysis of substantia nigra neurons, and then we saw that at this time, there's a down-regulation already of the genes related with transmitter release, with the synaptic function, etc., supporting this concept that the lack of dopamine is parallel, but a decrease in the machinery supporting dopamine release. And also we could demonstrate that directly because we could measure dopamine release directly in the axons here in the striatum. As you can see, a very clear, very marked decrease in dopamine being released in response to local electrical stimulation. And here, but we observe that dopamine release, dopamine in the nigrostriatal pathway, there has been a lot of attention paid to dopamine release in the striatum, but very little attention paid to dopamine release in the somatodendritic tree, okay? Here, when we measure dopamine release in the somatodendritic compartment, we didn't see any big change. Here, this is the quantification, which was, coincident with the fact that tyrosine hydroxylase, the dopaminergic markers were unaffected, okay? So, in parallel with this, we wanted to investigate what is happening with these neurons. As I showed you before, they have already axonal phenotype. What else happened? We look at how mitochondria are in this cell at P30, P35, when the soma is still working apparently fine, but the axons are already being affected, okay? Here, this animal, they show very altered morphology of the mitochondria. The cristae, where the oxidative phosphorylation, the mitochondrial complexes are located, are being already disorganized. And looking at this transcriptomic analysis that I mentioned before, we observe a general decrease in the genes encoding for oxidative phosphorylation, an increase in the genes facilitating glycolysis, a decrease in the inhibitors, an increase in the activator, and of course, a decrease in the knockout in the gene, which was downregulated, knockout. So indicating that these neurons are not using oxidative phosphorylation anymore. Mitochondria have been very much modified, and they are having a sort of metabolic remodeling, supporting the survival of the function of the neuron. And this can be demonstrated by looking at the ATP modifications, where ATP in the neuron is coming from. We measure cytosolic ATP by a perceval, which is an ATP fluorescent indicator. And in normal neurons, when you measure this perceval measures the ratio, ATP to ATP ratio, okay? When you look at where the ATP is coming from, if you add oligomycin, that it is an inhibitor, mitochondrial complex 5 of ATP synthase, you can see a decrease in ATP in these cells. And when you add to deoxyglucose, that is a molecule that is not metabolized, similar to glucose, but is not metabolized, you can also have a further decrease in the ATP concentration. And in this way, you can evaluate it, you can measure which percentage of ATP comes from oxidative phosphorylation and from glycolysis in these particular cells, okay? So what we saw that this is a normal cell, but when we look at neurons which are already after P30, P35, which are already in the early stages of Parkinsonism, you can see that the oligomycin doesn't do much, indicating that oxidative phosphorylation compatible with the transcriptomic data is very much gone, practically gone in these neurons, and the ATP synthesis is very much dependent on 2-DG, 2-deoxyglucose, indicating that practically most of these cells have undergone a metabolic reprogramming in which the deficiency of oxidative phosphorylation is being compensated by the increase in the synthesis of ATP by glycolysis. It's a sort of Barbour effect that also occurs in other cells, like, for example, in cancer cells. And probably this shift in metabolism allows these neurons to survive. And here, this is finally the quantification of this data, okay? The percentage of energy obtained by oxidative phosphorylation is about 50–60% in the normal animals, but in the knockout animal, only in the around P30, P35 is very much decreased. So, and also when we look again to the transcriptomic analysis in these cells, we saw big changes in the genes encoding for a number of ion channels, mainly those ion channels participating in pacemaking. It is very well known physiologically that neurons in the substantia nigra, in the normal condition, they fire action potential like a pacemaker. They generate about, in a very constant way, they generate about 2 to 5 to 6 action potential per second, okay, at a constant frequency in the normal cell. But in these neurons, which are already being affected, which are already fine, they are alive, but their axons are being affected and do not express dopaminergic markers. They are all silent, some of them, or they have a very abnormal spiking in which the frequency of spiking has very much decreased. This is logical because with every spike, you have the movement of sodium and potassium ions, and you need a lot of ATP to activate the sodium-potassium pump and the calcium pump to regenerate the gradient, the ionic gradient. So it is logical. This is viewed as a sort of adaptation to a more economical situation in these neurons, which are already with a limited amount or limited possibility of producing ATP in the cells because oxidative phosphorylation is not working anymore. Okay? So as time went by, okay, and we look at neurons later on at P60, P70, etc., we saw that the disappearance of the dopaminergic phenotype in the striatum progresses was much higher, and already there was a beginning, started to show a decrease in the number of Th-positive soma in the dendrites in the mesencephalon. Here you can see the quantification, a decrease in the number of neurons, and this phenotype was accompanied with, of course, a decrease in dopamine release already in the striatum, but also now in the substantia nigra quantitatively. And in this situation, again, this firing frequency, this phenotype, this economical phenotype was maintained. Which was interesting was when we, in these conditions in which the soma, there is a decrease in the number of mesencephalic dopaminergic mesencephalic neurons, when we inject fluorogol in the striatum to stain the soma, we can see still many cells which are fluorogol positive, and they are also Cre positive. There were cells that were before were Th positive, indicating that the cells lose the dopaminergic phenotype much before dying, because these cells still, they are alive, they still produce action potential, they can maintain the resting potential, etc., etc. They can be activated by synaptic input. These cells, which were before Th positive, but now they are not Th positive anymore, but they are still alive, indicating that they survive for several weeks. And later on, of course, these cells end dying. And when we measure with a marker, with a neuronal marker, we can see a very clear decrease in the number of neuronal bodies in the mesencephalon, in the substantia nigra. This is the last stage of surviving of these cells. So in review, what we have seen in this model is that the dysfunctional mitochondrial complex 1 dopaminergic neuron produce a progressive axon-first loss of function, mimicking the regional pattern of pathology in humans. Before we see the striatal pathology, then as the disease progresses, at least in our model, we see a somatodendritic pathology, lack of dopaminergic phenotype, and later on we see a neuronal destruction, neuronal death, okay? So, and this feature of our model allow us to study, to associate different behavioral statuses to different conditions in order to investigate how the correlation between the regional dopaminergic phenotype and the behavioral deficit. And a very interesting observation, I think, was that in the early stages, when we have only striatal axon pathology, we saw that these animals have alteration in associated motor learning and finer motor tasks. For example, if you study white maize here, these tests, the animal had to go to get food. The number of, in the wild type, the number of correct attempts is very high. The animals already lacking striatal dopaminergic innervation have failed many times, and this can be recovered with levodopa, but this is a striatal-dependent behavior, okay? But later on, when the disease progresses, at peak 60, for example, levodopa cannot recover because, this axon has become insensitive to levodopa, okay? This is very interesting because at this time, when you look at when you look at the behavior in the open field, the animals are completely normal. They move around. I think this is the knockout, okay? It moves even more, so they do not show any alteration in the gross movements, indicating that the alteration in the gross movements are not strictly dependent on striatal, sorry, I don't know how to stop this. Okay, it's not dependent on striatal loss on the loss of striatal phenotype. And here you can see what I showed in the video, the open field, the distance made by the animal in the wild type and in the knockout at P30 with only striatal denervation, and what happened later on when you have both striatal denervation and somatodendritic dopamine denervation, loss of striatal phenotype, and also loss of mesencephalic phenotype. And as you can see here, there is a decrease in the open field distance, okay, with time that can be also recovered with levodopa. And again, here, just showing in a moment, this video again, just for you to see that this animal, sorry, don't know how to move this thing well. Okay, it doesn't matter. This figure was just to show that there's two animals. This is the knockout. This one moves a lot, but this one doesn't move at all, indicating that after this kind of scheme. Okay, after losing the mesencephalic dopaminergic phenotype, there is the appearance of the gross alteration in ambulation, in bradykinesia, okay? So finally, we have a Parkinsonian model which teaches us, I think, give us information about how the progression of the alteration in the nigrostriatal pathway is affecting how neurons adapt to that metabolically and how this is affecting the behavior of the animal. Finally, we wanted to address this final question, is that, mitochondrial complex 1 has two main functions. One function is that, as I mentioned before, is NADH dehydrogenase activity. This is the basic metabolic function. But in parallel with this, for every molecule of NADH that hit dehydrogenase, there is a 4 proton that are transported. This is a proton pump through the inner membrane, generating the electrochemical gradient to generate ATP by mitochondrial complex 5, by ATP syntase. So we wanted to know which one of the two, whether the two or any of these two activities is critical for the Parkinsonian phenotype. And to address this question, we have done experiments recently in which in animals without mitochondrial complex 1, in which the two functions, the metabolic function and the proton pump function and the pump function are the bioenergetic function, I would say, are removed. We try to rescue the phenotype, injecting, with a genetically modified animal expressing with the transgenic expression of NDI1, which is an NADH dehydrogenase coming from yeast, which does not proton pump. It's a single molecule dehydrogenase, but metabolically does the same job as the 45 subunit mitochondrial complex 1. And in this animal model, this animal model is able to dehydrogenase NADH and to transfer electron to mitochondrial complex 3. Okay, in this animal model, as you can see here, we can rescue almost completely the striatal and the substantia nigra phenotype. So we have a very clear recovery of the histological Parkinsonian phenotype, and also looking at behavior. You can see here the gross alteration seen in this animal. I think this is done at P70 or P80. These gross alterations in the ambulation are also very much prevented, rescued by the expression of NDI1. And here you have the responses of this animal to the different behavioral tests, indicating that not only there is a histological recovery, but also a behavioral recovery. So in conclusion, I think that in perspective, in an animal, we think we have an animal model in which we show that the loss of mitochondrial complex 1 function in dopaminergic neurons is sufficient to produce progressive axon-first Parkinsonian phenotype, Parkinsonism, that is levodopa responsive in many aspects. This model is very similar to many of the aspects of Parkinsonism in humans. Okay, in this model, dopaminergic neurons undergo an adaptive metabolic remodeling, switching from oxidative phosphorylation to glycolysis. It's very important to know that with glycolysis, neurons can survive for months without maintaining action potential, synaptic activation, etc. So look like they do not require immediately mitochondria. It is unclear what drives the axonal and somatodendritic phenotypic remodeling and why the axons are more vulnerable than cell bodies. This is something that we have to study in the future. And also I think that this model may be very interesting to investigate the differential vulnerability of substantia nigra compacta neurons. It is known that the substantia nigra pars compacta neurons die progressively during the Parkinson's disease, but different neurons are affected in a different way. The ones that are very sensitive are the ones located in the ventral part and the ventrolateral part of the pars compacta, not expressing Cal binding, expressing the GRKK2 potassium channels, and expressing angiotensin II receptors, etc. So it would be interesting using this model to identify which neurons are affected first, okay? And also we show that rescue of NADH dehydrogenase activity without recovering the bioenergetic function of mitochondrial complex 1 with transgenic genes and NDI1 prevent the Parkinsonian phenotype. This is, I think, interesting because meaning that the cells with a limited amount of ATP, they modify their function, but they can survive, as I mentioned before, for a long period of time. This is very interesting because recently, just a few months ago, a clinical study, a randomized phase 1 clinical trial, appeared published in Cell Metabolism, in which Parkinsonian patients received nicotinamide riboside, which is used to increase the cerebral levels of NADH, so facilitating the action of mitochondrial complex 1. And in this phase 1 clinical trial, they already reported preliminary data suggesting very positive that this drug is very well taken by the patient, is very well affected, doesn't affect, there are not any negative outcome, but also they reported some preliminary data suggesting positive effects in the evolution of Parkinsonism. So then this may be correlated to the need of mitochondrial complex 1 to favor the NADH dehydrogenase activity in dopaminergic, in mesencephalic dopaminergic neurons, okay? Also, the staging of the deficit in mitochondrial complex 1 PARC model argues against the striatal dopamine depression is necessary, suggests that the striatal dopamine depression is necessary but not sufficient to produce clinical Parkinsonism. As I mentioned before, you have to lose the dopamine in the mesencephalon to really get rid of to really have the animal to appear really Parkinsonian, to see the bradykinesia, etc., etc. This is very important because so far the role of the somatodendritic dopamine release was described many years ago by a group in the Collège de France, by Glowinski, who was a neurochemist in the 70s, who was a paper published in Nature, but nobody paid much attention to that, and nowadays it's becoming more and more interesting. It looks like the somatodendritic release of dopamine is critical for the function of the whole system. How, how the network pathophysiology, I mean, the connection between the nigra, the putamen, the GPI, etc., how this is going to affect to the to the model that nowadays we have regarding the circuitry is something that will be studied in the future. And also we suggest, also our data also suggest that boosting dopaminergic signaling selectively in the mesencephalon rescues ambulatory deficit in mitochondrial complex 1 PARC model. This is very interesting because probably we should pay attention to increase the dopaminergic as neurons are maintained alive for a long period of time. If we add with some way, we activate the dopaminergic phenotype of in the mesencephalon, this may help to recover some, to rescue partially to Parkinsonian patients. For example, right now there's a clinical trial going on using dopamine decarboxylase administered by adeno-associated viruses in the substantia nigra to increase the amount of dopamine in these cells to see whether this improves the symptoms, okay? And this eventually it could be an interesting alternative to deep brain stimulation in the subthalamic nucleus that is currently used in people with advanced Parkinson's disease. And just to finish, I want to thank all my collaborators. This project, the mitochondrial complex 2 project was done in my lab years ago with these colleagues. The mitochondrial complex 1 PARC generation, this animal model with mitochondrial complex 1, was generated in my lab, and we did the initial studies. And also we have done recently the work with the transgenic expression of NDI1. Patricia Gonzalez Rodriguez, we collaborated with James Surmeyer, which is an expert in basal ganglion physiology, and she took our animal and went to his lab to do the full characterization of this animal model. They did a very good job in collaboration with Paul Schumacher, who is a friend also from Northwestern University in Chicago. He's an expert in mitochondrial bioenergetics and the rest of the group. And here you can see the whole financial support. And thank you very much for your attention. I'm going to go down, okay, to listen, otherwise.
Okay, questions? Ramon?
I get to get close to you, otherwise I don't hear you. I am sorry. Yeah.
Thank you very much. I think this is an extremely important finding. It changes a classical dogma of neuroscience that neurons are...
Speak without microphone. I don't understand, sorry.
I think it's an extremely important finding. It modifies a dogma of classical neuroscience that neurons are exclusively dependent on oxidative phosphorylation. I think this is very important, especially to identify possible treatments for Parkinson. And I wonder whether it's a mystery why these neurons survive so long with glycolysis. And I wonder if they try to differentiate. Have you explored this possibility? What's your hypothesis of these neurons finally dying? That would be extremely interesting to me.
I hope that you hear well. The question is whether the neurons that are surviving without oxidative phosphorylation do de-differentiate. And that's a good question because it is very well known that oxidative phosphorylation, when you study the mitochondrial dependency of neural stem cells on mitochondria, you need mitochondrial function to produce neuronal differentiation. Okay, that is a good question. Could be, but so far our data show that you can have neurons which have lost already the dopaminergic phenotype and they fire action potential beautifully. With some modification, I didn't go into details, but with some modification, but they can fire action potential repetitively. They can be activated by cage glutamate released nearby the somatodendritic tree. They produce a huge postsynaptic current and producing a huge activation of the neuron. So they maintain the glutamatergic synaptic contents, etc. So the indication, they maintain the shape, the morphology. Your point is very interesting. It should be taken care of, course. It should be thought about it. But for the moment, in the time frame that we have studied these cells, look like they die before de-differentiation. Okay, because at the end they die. Okay, that's important to know. Okay, although they can survive for many months only with oxidative phosphorylation, mitochondria are needed. Okay, for survival. Okay, but it's okay.
It's a very, very nice study. Congratulations on the beautiful data. I was wondering, do you have an explanation for why the axonal ramifications, the axonal functions decrease first before the actual somatodendritic functions end? Is there any way you can possibly test this?
Yeah. Well, I think that is a good question too. We think that probably, in the axon we have all the exocytotic machinery that you know, of course, very well. This machinery needs energy somehow the axon to maintain the gradients, etc. They need mitochondria, but the machinery, as far as I know, we don't have much ribosomes in the axonal. Although there's some ribosome, but not many ribosomes in the axonal terminal. So a possible explanation could be, at least one I have been thinking about it, is that you can get they have to be synthesized in the soma and to be transported to the periphery. Correct me if I am not right. Okay. So if you are having the neuron not working well, and you want to spare energy, or you spare energy than regulating the ion channels responsible for firing, okay, for pacemaking, and also decreasing the amount of protein, which is costs a lot of energy to synthesize them and to send them to the periphery, axonal transport, etc., etc. That could be a possible explanation, although maybe there are others. That could be a reasonable explanation. Okay.
And that will be the last question.
Yeah, that's it.
Are you thinking of...
I'm very sorry, but I am very deaf. I have my earphone here, no, but they don't help.
So are you thinking of looking at astrocytes in the neighborhood? Maybe they are also having some metabolic remodeling to further support neuronal survival?
I don't remember. I was not personally every day involved in the experiments that were done in Surmeyer's lab, okay? But I think they look at astrocytosis during this process. They didn't see much, only at the very end. They saw a little bit of astrocytosis. There are very few astrocytes.
You don't need to have any astrocytosis. Just metabolic remodeling, meaning that the lactate shuttle or whatever is...
I mean, metabolic remodeling in astrocytes.
Right.
We cannot look at that. Our knockout does not affect astrocytes, in principle.
Yeah, but as a secondary response...
Okay, but we cannot look at that. Okay.